Hello guys, today I will show you analysis with GAGE, pathway analysis, how to do the pathway analysis with GAGE. So I'm using mouse data. This is single cell sequence data set. So what we are gonna do, we're gonna start trying to find the difference between wild type hypoxia and wild type normoxia samples. So of course, Surat is the number one indispensable package which is used for single cell RNA sequencing. You will use this library, and also you need the library of mouse data set, which is org, mmeg, db, and also you need library of gauge data. I will show it later. So not to forget what I have done so far to analyze the data, I am trying to record it to YouTube. So first, of course, we are going to upload our RDC Surat object. I have already done it. So first, I'm going to find the DEGs, differential express genes. I'm doing it with find markers function of the Surat, where one is wild type normoxia and wild type hypoxia. And after that, I'm going to filter these differential express genes based on p-value adjusted smaller than 0.05. And also, I want either PCT1 or 2 to be 0.1, which means 10% of cells required to express it. So after I filtered the DGs, then I'm going to change all of these DGs, significant DGs, differential express genes, to the anthracite symbols, okay, from the normal gene symbols to anthracite. Okay, anthracite, not another symbol, okay, doesn't matter. So after that, you will take these anthracite names and you will change with the significant DGs row names, and then you will filter more the anthracite. Why? There are some NA values, so you don't want any NA values in our data set. That's why we are only getting address valid values. And after you get this valid value, also you will filter your significant DGs according to these valid interest values. And after that, you are going to find expression. You are going to make a new data frame. This data frame only includes your genes, significant DGs, and then also your fold exchange. And then you will start using library gauge data, where you are going to retrieve key pathway genes for mouse, and then you will start your function. Your function is gauge expression data is passed here, and then your data set here, and then you don't need to use reference or sample values because you already combined those values in expressions here. So then you will compare this data set. After you get gauge results, you will make it as a kind of a data frame, and then you will take the first 20 based on the stat mean. Stat value is a kind of value. If it's positive, it shows this pathway is upregulated. If it's negative, this pathway is downregulated. So based on what you want, you're gonna do it. So after that, it's time to visualize it in your top DF, your data frame, and then you will reorder your data frame based on the highest stat mean. And this is ascending manner, by the way, but depending on where your row names are shown in the graph, you can adjust it. And then you will get the ggplot. So if you don't want to do it again and again, you can make a kind of for loop. First, write all the pairs in a list, and then use a for loop. Ident 1 is pair 1, ident 2 is pair 2, and then use this DG and all the other steps based on these pairs, and then you just save all the data set in a folder. That's simply how you can do it. And I think that's it. Thank you for listening to me.